Okay, so let's start. I'm Hofmeyr, and would like to introduce Stephen Hofmeyr. I hope I spell it correctly.
Close enough.
One of the lead developers for MultiAssembly, and he will give us some introduction today. And Stephen, your stage is yours.
All right, thank you, Hofmeyr. Right, so I'm going to talk about MultiAssembly, which is a meta-genome assembly application written in UPC++. So to give you a bit of background, I work at the Berkeley National Laboratory, which is based in California. And MultiAssembly is developed as part of this thing called the Exascale Computing Project, which has a number of different projects under it, and MultiAssembly is part of the Exabiome project, and I'll talk a little bit more about what that's all about. The original MultiAssembly version 1 was released a number of years ago, and that was written in UPC, which you can kind of view as a precursor to UPC++ at MPI. And then we've recently released version 2, which is entirely rewritten in UPC++, which is developed by another ECP project also at LBL. And the new version runs a lot faster and uses less memory than the old version. And this is thanks to some degree to UPC++. So let me just give you, I'm going to give you an overview of what this is all about. And what MultiAssembly does is meta-genome assembly. So the idea behind meta-genome assembly basically is that it's not like, when you're doing normal genome assembly, you would take, for example, a sample of an individual or an animal's genome, and then you would try to assemble that. What you do with a meta-genome assembly is you take an environmental sample. So, for example, you'll take a scoop of soil or a cup of water from a lake, and that environmental soil will contain microbial genomes. Tens of thousands of microbial genomes, all mixed together. Some are very common, so they have high frequency. Some are rare and have low frequency. And then the way the sequencing, the genome sequencing works is the only way you get to read the DNA of all these organisms is by breaking it up into small fragments called reads. And a typical read length might be about 150 to 250 base pairs, where each base pair is A, C, G, or T. It's one of the letters of the DNA alphabet, as it were. And so you don't get a lot of contiguous information when you do the sequencing. And you also have some small level of errors. It's about 0.25% per base. And in order to deal with the errors, you don't just get one copy of the sequencing from the environment, you get multiple copies. And this is called the sequence depth, in case you've heard of it. So the number of reads that will map to a certain genome depend on both the depth and the frequency or abundance of that genome. So that's all, as you can see, the sort of top two areas of this slide here are all what happens with the sequencing technology. And then when you get to the assembly here, it's all passed to a computer to do. So basically the idea behind the assembly is you've got all of these short reads, as I said, 150 to 250 base pairs long, and you've got to computationally connect them together to form longer sequences called contigs or contiguous sequences. And you want to do so in a way that minimizes the errors and makes them as long as possible, because the longer they are, the more biologically relevant information you can extract, like complete genes and things like that. So that's basically the problem that's addressed by MultiAssembly. Let me just give you a little bit of, I'll dive into more details about MultiAssembly later, but let me just tell you a little bit about what the usefulness of meta-genome assembly is. And it's very helpful for science analysis of a number of different environments. And here's just a sampling. MultiAssembly is actually used currently by the Joint Genome Institute, which is a department at LBL, where they do a lot of large-scale sequencing and genome analysis. So one of the things you can do with meta-genome analysis is you can, for example, try to understand what happens to microbes after a wildfire. Or you can look at soil carbon cycling. And soil carbon cycling is obviously of increasing importance as we confront climate change. We also know that microbes can affect disease in various crops, and so we've got projects that look at the how they affect disease for the growth of switchgrass, and switchgrass is something that's being investigated as a biofuel. We can also look at the impact of the various microbes on mangroves and other sort of environments, see how they change over time. And we can also do further downstream processing, such as isotope tracing, to try and improve our understanding of how these things are used. So one of the interesting things about MetaHitmer compared to other kinds of metagenome assemblers you can see out there is that it runs on distributed memory system. So we can run this on a supercomputer, and it scales well on a supercomputer, and I'll give you some data on that. What this means basically is that we can assemble very large data sets, because one of the main constraints is not just time, but it's memory. So you need a multi-node distributed system with lots of memory to assemble the very large data sets. And here I'm showing you just a snippet out of a study that we published recently in Nature Scientific Reports. But basically what we're doing is we're comparing a couple of different approaches to assembling a metagenome. So the approach that used to be used for these very large data sets with the existing assembly tools is something called MultiAssembly. So basically you take your large data sample and you split it up into lanes or different subsamples, and you assemble each one separately, and then you somehow combine them. You concatenate them, and then maybe you deduplicate to try and remove duplicates. What MetaHitmer allows you to do is it allows you to co-assemble all of these samples together. So you run one assembly with all of the samples together, which obviously requires a lot more memory. But the benefits are huge. You get much longer and less redundant assemblies with a lower rate of errors. And that's what this plot tries to show. On the y-axis, we have the cumulative length in base pairs. So that's the number of characters you've assembled. And on the x-axis, we have the number of contigs, which are these contiguous sequences. So here we have, this plot is ordered from the longest contiguous sequence to the shortest. And as I said, it's a cumulative length. So if you read the y-axis, this is a log scale on this plot here. The very first starting point, 10 to the 0, is obviously 1. That's the length of the longest contig that we've managed to assemble. And in this case, that's like, I don't know, maybe 200 million in length, something like that. And you can see by looking at this blue line that you're assembling much, much longer sequences than you are with CoAssembly, which is the blue line, than you are with MultiAssembly. So this has several benefits, and I'll just talk about one of those, is that a CoAssembly allows you to find rare genomes that you would never find otherwise. Because basically, if you take a sample, if you take a big data set and you split it up into subsamples and you assemble each one individually, you get very small fragments of the low abundance genomes. And so it becomes very difficult to connect those together. You just don't have enough information. Whereas if you do multi-assembly, you can potentially connect those. And so what we're showing here in this graph is how we can find rare genomes. So on the y-axis here is the fraction of genome that's been recovered. It's the percentage, and the higher the value, the better. And on the x-axis is the sequencing depth. And the sequencing depth is related to the abundance of the organism. So what we've done here is we've injected some synthetic organisms into the data set to see at various abundances to see how well we recover the genome fraction. And you can see here, if you look at the blue crosses, this is CoAssembly. Even as the sequencing depth or the abundance of the genome gets low, we're still doing a very good job at getting most of the genome fraction. When we have the lowest sequencing depth, then our genome fraction drops off, obviously. It's got to drop off dramatically at some point. But you can contrast that to MultiAssembly, which is these little golden pluses here, which drops off much more abruptly. And so basically with a sequencing depth of less than 40, you can't do anything with a multi-assembly. And one of the reasons why this is very important is that rare genomes are often of more interest in a sample than the common genomes. The common genomes, abundant genomes, are often well known because they're abundant. So if you're trying to find out new species, then you need to know what the rare genomes are. So that's just a bit of the biological overview and motivation. I'd just like to say a few words about the programming framework and that MetaHitmer2 is written in, because this is not MPI and has nothing to do with MPI. And so I think this strikes a contrast to the other applications you've been dealing with, and it sort of offers different challenges for performance and tuning. So MetaHitmer2 is written in UPC++, which is basically a C++ library that allows you to run on a parallel program on a distributed memory system. It has support for one-sided communication, RMAs, so you can do remote puts or gets to a global memory. It also has support for remote procedure calls, where you can ship your computation to where the data is. It's by default, everything is asynchronous, so it becomes easier to overlap computation and communication, and it has support for collectives and several other features of that you would require in a distributed programming environment. It runs on a communication layer called GasNet-X, and this is a low-level layer that provides a network-independent interface to UPC++, and it runs on several different hardware platforms. GasNet-X itself is used by more than UPC++. There's quite a number of different programming environments that use it, and it's been around for years and years. So as I said, neither of these systems use MPI. They are interoperable with MPI, as we've seen people using MPI with UPC++. Also, MetaHitmer, just to clarify things, is a sort of a process-based, uses UPC++ as a process-based model. It doesn't use OpenMP at all, so it's not hierarchical parallelism, which is not to say that you can't interoperate OpenMP with UPC++ and GasNet. You can, but MetaHitmer doesn't use it. So just one caveat is, depending on whatever system you're working on, the job launcher may use MPI run or something like that to launch every application, including MetaHitmer. So what's the benefit of using something like GasNet-X? Well, one-sided communication can be a lot faster than two-sided, which requires not only more programming effort potentially, because you have to synchronize and coordinate the send and receives, but there's also more communication required because of round-trip requirements. So what we're showing here is a performance comparison on two different systems. We have similar plots for for more systems than this. On the top, you see Summit, which is one of the fastest systems in the world. It's at Oak Ridge, at the Oak Ridge Computing Leadership Facility. And at the bottom one, you see running on Cori, which is at NERSC. It's also a very petascale system. And what we're showing here is the flood bandwidth, point to point. So the higher you are on the x-axis, this bandwidth, the better the results. And on the y-axis is the size of the transfer you're making. And so you can see, for example, on Summit, this blue line here is GasNet put. So you're putting memory, you're putting to a remote memory location, and the red line is GasNet get. So you're getting from a remote memory location. And you can see that this top line here is the GasNet performance. The circles here are the same thing for MPI, has one-sided puts and gets as well. And these are the same thing for the MPI one-sided puts and gets. So in this range on Summit, GasNet considerably outperforms MPI. For example, here it's about twice the bandwidth. If you look at another system here, we're looking at the same thing, these circles versus the crosses. You can see that GasNet is consistently outperforming one-sided MPI. And then two-sided, which is how most MPI applications are currently written, is much, much worse performance in terms of bandwidth. So MetaHIPMA 2 is not a typical scientific application. It has very different computational motifs from your normal scientific application. The most common computational motif is a distributed hash table, and they happen to be everywhere. And these hash tables have basically random global memory access with very little locality. The communication tends to be asynchronous, irregular, and point-to-point. We still get good scaling because this kind of we can achieve good load balance with this kind of system. And the way the hash tables are typically used is we typically generally do all of the updates first and then all of the accesses. So you put a whole lot of information in the hash table and then you use that information. One example is graph traversals, which we do a lot of graph sort of manipulations in this assembly, and I'll talk a little bit about this in this application. I'll talk a little bit about that later. But for example, with the graph traversal, what you would have to do is you first build up the graph, and each location in the graph is basically a location in the distributed hash table, and they can be scattered throughout the network. And then when you've built up the graph, then you can do things like traverse it by basically following the links in the graph and accessing the information from the distributed hash table. Another key point about MetaHIPMA's computational motifs is that it has hardly any floating point, and there's no floating point in the critical path. It happens to be mainly string manipulation. And the strings are sort of tend to be packed variants of the genetic code, the ACGT. And it happens to be, as I said, very memory intensive. We have, for example, with large scale data sets, we have a minimum number of nodes that need to be used simply because of the memory requirements. And these characteristics make it pretty challenging for GPU optimization. So what you are going to see with your use of this application, with your tuning of this application, is we're not using GPUs at all. But we have been working on adding GPU optimization to this project. And we believe we can get some performance improvements, but not dramatic ones like you would get with other applications. So the MetaHIPMA application is basically an assembly pipeline, and it's a fairly complicated sort of pipeline, as I'm indicating in this plot here, in this figure here. So you start off the pipeline with a bunch of reads, and I said that these are strings of DNA that basically came out of the sequencer, and they are 150 to 250 base pairs in length typically. And you have multiple layers of these reads basically, and that's the sequencing depth I talked about before. So each read can cover potentially each part of the final genome can be covered by multiple reads. So you have to combine those multiple reads. So the first thing we do with these reads is a process called Hofmeyr analysis. We take the reads and we split them into short fragments, typically 21 to say 99 base pairs in length. And these fragments are called Hofmeyr's, where K is the length. And what you do is you split them into these fixed length Hofmeyr's, and then you create something called a brain graph, which is basically a graph where the nodes are, or the vertices are, each vertex is a Hofmeyr, and the edges between the vertices are determined if there's an extension that goes from one Hofmeyr to another Hofmeyr. And using this brain graph, you can then do contig generation, which is the second phase here. So the first phase, you're basically creating this distributed hash table and you stick all these Hofmeyrs into it. And then the second phase, you walk this distributed Hofmeyr graph. And what you're looking for is trying to connect all of these short Hofmeyr's together in as long a walks as possible. And so if you you would look at a Hofmeyr, which is at a vertex, and you would look for the extension, and then using that extension, you know what the next Hofmeyr could potentially be, and you'll try and get that one from the graph. Now, if there's multiple possible paths, then that's a fork, and you can't at this point decide between it, and so you have to terminate your walk. If there's no new path, then you have to terminate your walk as well. And so out of that, you get these contiguous sequences or contigs. which are longer than the original k-mers and hopefully longer than the original reads too. Then we do a stage where we take all of the original reads and we align them to the k-mers. And alignment is a very common operation in bioinformatics. It's probably the most common operation. But basically, you try and find out how those reads line up to the context. And once you've got a collection of reads that line up to a context, you can use the information in those reads to just independently extend the ends of each contig. And this is the local assembly stage. And then once you've done this whole process, it gets iterated, and then there's another loop. And you keep iterating this loop, and at each stage, you increase the value of k, or the length of this k-mer. You start with a low length, and then you increase it with each iteration. And typically, a run might have like 5 iterations. And you can see that when you run MetaHipmer in the output, it's informing you that it's done multiple contigging rounds. Each one of these is a contigging round. Now, the reason we do these multiple rounds is that in order to capture any information about low abundance or low depth genomes, we need to have short k-mers, because the longer the k-mer, the more possibility there is that there's an error in it, and so we can't use the k-mer. Because what I forgot to mention here is when we count the k-mers, we drop all the k-mers that occur only once. We assume they're errors. They have to occur at least twice. So if the k-mer length is longer, then it's much harder to get information about the rare genomes. But if the k-mer length is short, then it's hard to it's hard to make a contiguous sequence over repeat regions, because you get a lot of forks in the graph. And genomes are characterized by repeat regions, or basically loops in the way the genome is constructed. So the longer k-mer lengths help you overcome repeat regions, and the shorter k-mer lengths help you recover less abundant genomes. And so that's why we iterate through these loops. And at each stage, we take the contigs that we used, that we developed from the previous stage, we split them up into k-mers themselves, and we add them into the mix. And at the end of that contigging loop, we've done a number of contigging phases, you end up with these extended contigs, and then you do a whole another phase where you basically try and link those contigs together in scaffolds. And that's, I've simplified this stage quite a bit. That's another complicated stage. But basically, you're treating this where every contig is the vertex in the graph, and the edges between the contigs are determined by what reads map onto the contigs. And then you have another graph that you walk, and it's a different kind of graph to the brain graph. So what does this these different stages look like in reality when we try and compute them? And I'm going to show you here how long each stage takes relative to the others. So these results are for doing an assembly of an 820 gigabyte data set, which is something you wouldn't do unless you had a sufficiently large memory. And this is on NERSC Cori supercomputer, the Knight's Landing partition. And what we're showing here is, oh, I see. This is, I'm missing some information here. This is Cori Knight's Landing, and this is the Summit supercomputer at Oak Ridge National Laboratory. And the Summit supercomputer has, I think it's like half a terabyte per node memory, whereas these KNL nodes only have 96 gigs memory per node. So you need a lot more. Here's the number of nodes on the x-axis. You need 256 nodes at least on KNL to assemble this. On Summit, you only need 64 nodes because you've got more memory per node. And then this is the time taken to do the assembly. So obviously, the smaller this number, the better. The overall height of the column is the overall time taken for the assembly. And then the colors within are the breakdown between the various stages. So, for example, this color here is file IO, which looks very similar to k-mer analysis, but basically you can't see it in any of these plots. So file input output on, at least on this supercomputer, the cost of IO is pretty small. It's obviously using a distributed file system that's high performance, so that helps a lot. But you can see the various stages. For example, k-mer analysis tends to be the largest scale, the stage that takes the most time. Then there's alignment and local assembly as well. And the breakdown differs somewhat between the different architectures. But one thing you can see here is that we're getting pretty decent scaling. When we go from 256 to 2,000 nodes, we are getting a decent sort of speed up. And it's more pronounced on Summit. The exact breakdown of how these different stages, the amount of time these different stages take, depends both on the data set as well as on the machine. So, for example, if you have a higher depth data set, then you will have increased Hofmeyr analysis time, and the things like alignment will decrease, relatively speaking, and scaffolding will decrease. And then the machine, as far as the machine goes, a very important characteristic is a high performance interconnect. So if you try and run this, this application on high-speed Ethernet, chances are using 4 nodes will be a lot slower than using 1 node. We've tried this, for example, on the Amazon EC Cloud, and we've seen slowdowns of potentially an order of magnitude going from 1 to 4 nodes because the interconnect just simply wasn't fast enough. So it's very dependent on communication, this application. And sort of speaking of that, here's a little overview of what the communication patterns look like for MetaHIPMA. So this is once again our 820 gigabyte data set, and I'm showing the plot for 512. Actually, I think this is Summit nodes, not KNL nodes. This is Summit nodes. And what I'm showing on the left plot here is the volume of communication. If you look at the y-axis, this is the megabytes per process. So the higher this is, the more communication is happening. If you look at the x-axis, this is the communication each stage is engaged in. So this is the Hofmeyr analysis, that's the content generation, that's the alignment, local assembly, scaffolding, and that's the total. So you can see that the most communication volume happens in the Hofmeyr analysis. Alignment is quite high. But what this kind of communication volume plot hides is the fact that there's very different message sizes that are used in the different stages. So for example, and this is what we're showing here, the average message size, which is on the y-axis, and then once again the stage here. So in this example here, we can see that even though content generation has a lot lower communication volume than Hofmeyr analysis, messages tend to be very small, because what you're doing is you're traversing this graph and you're fetching just little bits of information about what the next vertex is. And because of that, this content generation stage tends to be more latency bound than bandwidth bound, whereas something like Hofmeyr analysis and alignment are more bandwidth bound. So what we're getting basically is these two typical patterns of communication. We get, and they're always random point to point, but you either get small active messages, such as this graph traversal or content generation I was talking about, or you get aggregated messages, such as the Hofmeyr analysis, which are larger. And once again, the number and size of the messages depends on the data set and the number of processes and nodes you're running on. Because if you are running a large data set on not so many nodes, then you can do a lot of aggregation, because each process will have more data that is destined for remote rank, so it'll be able to send bigger chunks at once. If you have more nodes, then you'll have smaller message sizes. So this 512 nodes is fairly large for this size data set, so you don't expect very large message sizes. So how does MetaHIPMA scale on very large data sets? So here I'm showing results of something we did recently, where we assembled an almost a 7.7 terabyte or almost 8 terabyte data set, and this is terabytes of reads. So this is the biggest data set that's ever been co-assembled, and it's the biologists are busy looking at the results and starting to discover some interesting stuff about it, because they can get information from this assembly that they couldn't get otherwise. But basically what this plot does here is it compares the performance on two different systems with different hardware. So what we're showing on the y-axis here is the size of the data set. And this is basically achieved by taking the 7.7 terabyte data set and assembling just one chunk of it, and then assembling two chunks, co-assembling two chunks, and then three chunks, etc., all the way to co-assembling the whole lot of it. And on the y-axis, we have the node hours taken to do the assembly, and we're using this metric in particular because we can then compare the different systems like Cori KNL with Summit. And so obviously and this is these are both logarithmic axes. The lower the node hours, the better. The more you desire the result. And so these green triangles, just sort of for historical reference, are assembling the data set up to about 3.5 terabytes on Cori, the NERSC system, with the old version of MetaHitmer, which was the UPC MPI version. And you can see that takes a lot more node hours than MetaHitmer 2, which are these two lines down here. And here the two most important ones are Cori KNL. So that's the NERSC system, the Knights Landing partition. We can see that as the data size increases, the node hours increase in a pretty sort of consistent way. And what we can also see here is the square markers are for Summit, and we can see that Summit is considerably faster than the KNL partition of Cori. We have one data point for the Haswell partition of Cori, and it's closer to Summit, which is sort of to be expected. So one of the things we can use this kind of plot for is to extrapolate how long it would take, at the very least, how many node hours to assemble larger and larger data sets. So here, we've assembled up to an almost 8 terabyte data set. Our eventual goal of the Exabiome project is to assemble 50 terabytes. And the whole exascale computing project is all about finding applications and technologies that are suitable to run on exascale systems. And so our challenge goal here, this co-assembly of 50 terabytes, is something we've chosen to be fairly suitable to an exascale system. And you can see that here because Summit is not an exascale system, but it's a pretty high petabyte scale system. And just extrapolating this linearly indicates that to assemble 50 terabytes, we need the whole Summit machine for an hour to do it. And it's very unlikely that we would scale, so well up to that number of nodes. So because I think this is like maybe 1,000 nodes or something like that. So in order to do the scaling of 50 terabytes, we would certainly need something faster than Summit. Some information that's relevant to anyone using MetaHitmer for this competition is all about tuning MetaHitmer. I don't want to go into a great deal of detail about this because there is a lot of guidance on the web page, and you can always obviously ask questions about this and what you can and cannot do. And I'm sure you all have the link, but I've just put it here in the slides just in case. The basic usage and the default usage of MetaHitmer is all you should need. You just run the Python wrapper script, and you pass it the -r and the file name. You will note that the competition file name is a zipped file, and you need to unzip that. MetaHitmer does not take zipped files as input. Then MetaHitmer can run, and when it runs by default, if you're running on multiple nodes, it checkpoints the results. So periodically, it saves everything to disk. If that's generally of use if you're running a very large assembly, or if you suspect your system may crash. In the terms of this competition, we don't need to do the checkpointing at all, and it will slow things down, and it'll make things more dependent on the I/O system, and we don't really want that. So just always specify no checkpointing. There are some parameters to MetaHitmer 2 that can be changed, and I'll detail a few of these down here. Your mileage may vary. It depends a lot on your setup of your system, whether these will help or hurt you. But let me just go through them a little bit. There's this parameter here, max K-mer store. So basically, when you are doing the K-mer analysis, where you splitting the reads up into these short fixed length sequences called K-mers, and you are deciding which ones get dispatched to which parts of the distributed hash table, these K-mers are aggregated. So basically what's happening is each process is accumulating all of the K-mers that are destined for each other process before it sends off a batch of them. And how much space it has to accumulate them is dependent on this K-mer store. And so we have some way of automatically tuning this. Normally you don't necessarily have to set it at all, but you can play around with increasing or decreasing the size of the K-mer store. It also depends on how much memory you have available. So typically, if you have more memory available, increasing the size of the K-mer store could potentially help. The next parameter is the maximum RPCs in flight. So the way the MetaHitmer is working is a lot of the operations are these remote procedure calls, and they are done asynchronously. So they're dispatched, and you keep each process that dispatches a remote procedure call keeps on working, and so it keeps on dispatching remote procedure calls. And the remote procedure calls basically can accumulate at the target and need to be processed one after the other. And if too many remote procedure calls are dispatched, then you can run out of memory. So what we do is we have a constraint on the number of RPCs that you can have in flight at any time, and that's this max RPCs in flight. And once again, we sort of tune this to adjust for the memory of the system. Generally, the more you can have in flight, the faster the system will go, but then you can potentially run out of memory. So you can play around with that parameter. For both of these parameters, you should be able to look at the output of MetaHitmer and see what it says about how much data it's using for the K-mer store, or how many RPCs it has in flight, and use that as a guideline to try, and how much memory is being used, and use that as a guideline to try and adjust the values up. One of the things you can do to get extra information is you can run this command with the -v for verbose, which will give you added information about what's going on. You can also look at the output directory. If you look at the, once MetaHitmer 2 has run, if you look at the end of the output of, the end of the text that's output when it runs, it will tell you what output directory it stored all the results in. And within that output directory, there's a file called MHM2.log. And that basically is an output of this run with the verbose mode on. So you don't necessarily need to make this verbose. You can look at that log file to see, get more information. So another parameter here is the pin parameter. And basically this pins the processes to cores. And we've got a number of different options for doing this. You can just not pin the processes at all. You can pin the processes so that each one is assigned to a pair of hardware threads, or you can pin it so that each one is assigned to a single hardware thread. There's several, or each one is assigned to a NUMA node. There's several different options. So you can play around with that pinning option. Another thing you can play around with, just thought of it, that's not, that I haven't, oh, actually it does come later. Okay, I'll talk about that in turn. So the next parameter here is the shared heap. This is how much memory is required for some of the shared memory data structures. Generally, you don't need to play around with this. Increasing it or decreasing it won't change performance, but there is a certain minimum that's required. And the greater the number of RPCs in flight, generally the larger the shared heap you'll have. So that is a value that sort of interacts with the rest, but don't expect any performance changes from just adjusting the shared heap on its own. And then the final parameter you can also play around with is prox, or the number of processes MetaHitmer 2 is running. By default, it will detect the number of cores in your system and run the same number of processes as there are cores. So by default, it doesn't use hyperthreading. You know, if there's two hardware threads per core, it's only going to use one process per core. So you can change that by increasing the number of procs, and you can experiment to see what gets you the most benefit there. And so this link is also on the web page, but basically you can look at our guide for more information about the various parameters. And as I say, these are the only ones that you really should be playing with. You should leave the others alone, because the other parameters may well affect the quality of the assembly, and we don't want the quality to be changed. I'll talk a little bit more about that in another slide. Then UPC++ itself and GasNet has a whole, they have a whole bunch of options, and you can look at the GasNet documentation. You can see that over here. And once again, it's also documented at the competition webpage. But there's, especially if you're running on InfiniBand, which is, I think, the network for all of the systems we have available, there are a lot of different GasNet options to play around with that may affect improve the performance of some stages, but not others. There's quite a bunch of scope for tuning there. And so this is the results of MetaHitmer. It's important to note that the yes, and I've actually written this slide wrongly. My apologies. The output of MetaHitmer, I'll correct this, is non-deterministic. It's not deterministic. It's non-deterministic because of the way the updates work. But the overall statistics that you get for a run should not vary significantly from one run to the next. And we actually have tools to do, and I think this is documented on the competition webpage. We have tools to do very close checking of the results. But the results, but you can, without even running the tools, you can eyeball the results. At the end of a run, it'll print out some of the assembly statistics, and this is an example printout here. And these shouldn't vary dramatically from one run to the next. The contact lengths that are found, the average contact depth, the maximum assemble length, these kinds of things should not vary dramatically. So when you're tuning and doing repeated runs and tuning the system, you should, and look at the web page to get the tool, but you should periodically run a tool to see that the results are the same, but you can just eyeball these assembly statistics to make sure you haven't changed anything. Now, the assembly statistics should not change significantly if you don't fiddle with, they'll only change significantly if you fiddle with the wrong parameters to MetaHitmer. If they do appear to change significantly without changing any of those other parameters to MetaHitmer, but changing just the ones I recommended, I'd like to know about it, because it means that there's potentially a bug somewhere. And that is my summary. So it's time for questions.
First, see the chat. There's a question from
yes, let me just, sorry, I had to, have I stopped sharing now?
Yeah, it's okay.
Yes, I had to stop sharing in order to see the chat. Let's see. Chat. Okay. Does GasNet X have a profiler or a tracing tool? Are there... GasNet X, so any profiling tool that is usable with C++ can be used with MetaHitmer. GasNet doesn't have an explicit profiling tool, but you can do tracing with GasNet. Sorry, MetaHitmer. So GasNet can trace communication operations. If you look at the documentation, you'll see there's a sort of a trace. I think there's an environment variable called trace options that allows you to gather aggregate statistics about the messages sent and the message sizes. It also will gather information about the number of puts and gets, and it can go into much more detail than that, I think. So take a look at the GasNet documents about that. So what's this? UPC has UPC XX VTune. I'm not sure if that's a question or not about VTune. Yeah.
I
So
think it's an answer.
oh, it's an answer. Oh, I see. Okay. Yes.
I think.
Yeah. Let me know if there's any other questions. You don't have to only ask questions
Yeah.
in the chat. I can.
I would mention something that basically the PGAS, GasNet, and they can run on top of verbs, right? InfiniBand
Yes. Yes.
verbs.
Absolutely. Yeah. Yeah.
And
That's one of the
possibly
skills.
also over MPI, but this is less, this performs less. So,
Yeah. Yeah, that's a very good point. GasNet has native support for InfiniBand, and that's where you're going to get the most performance. The performance will, if you're not making use of the native InfiniBand support, it's likely to be very poor compared to InfiniBand.
So basically, you can, if you're not familiar with PGAS or GasNet implementation, it replaces MPI. It can it's in a way another layer of message passing interface, another implementation of parallelism.
Right,
Okay.
right.
And for InfiniBand, it just works over verbs, which is the API, which is InfiniBand API. And
yeah, as I said in the slides, you should
yeah,
not have to use MPI. You may have to use MPI run to launch a job, but there should be no reliance on MPI for communication. You'll see in the GasNet documentation, it usually talks about numerous different conduits, network conduits. For example, there's the SMP conduit, which is used on a single shared memory node. That's the most efficient symmetric multiprocessing. You won't be using that if you have multiple nodes. There's also a UDP conduit. That's something you would use on high-speed Ethernet. It's low performance. It's something to be avoided if you can. And then there's the native hardware conduits, such as the InfiniBand verbs or Aries, if you're on a system like that. And that's what you want to preferentially use. So you want to make sure you are running GasNet using that.
okay. I will also mention that we have two clusters. One is the Niagara cluster from University of Toronto that team captains already should have gotten cluster access. So look for the email from the Niagara IT. And another cluster is the SCC cluster, is the cluster we had last year. And you should be able to use that as well. I want to mention another point about the GPU usage.
Could I just interrupt
go.
you and say both of those clusters, I think, are InfiniBand. Is that right?
Yeah, both of those clusters are InfiniBand.
Yep.
Slightly different CPUs. So the Niagara is slightly newer with the EDR and Skylake nodes. So 40 cores per node. And the NSCC is slightly older, one or two older generations, I think 28 cores per node or something like that. Also EDR, but ConnectX 4. So I want to mention also about the GPU usage. We decided not to use it, right? Because it's not well, it may not well debugged, right? It's not was not done great.
The version that you have for the competition should run fine on GPUs. The only thing, though, is that in that version, the amount of computation done on GPUs is generally very little, especially at scale. So it's not really, doesn't really make a big difference. So we sort of elected to exclude GPUs from this because it's a lot of, I mean, you could spend a lot of time trying to tune the usage of GPUs for very little reward because it isn't a very big part of the computational pipeline.
Yes. So also another thing to mention. I see another question. Is that possible to migrate some of the logic like DHT on GPUs?
It is, and actually we're busy working on that, but it's quite a challenging process because a lot of this is sort of, as I say, random point-to-point memory access, which is not the sort of computation pattern that is amenable to GPUs, but we have preliminary distributed hash table implementation on GPUs that does give us some speed up. So we're working through that. If this competition was happening next year, I would say we'd probably be using GPUs because a lot of this work would be happening on GPUs. It's worth noting that, you know, as we're talking about GPUs, that all of the exascale systems that we're sort of tasked with eventually running on are going to be heavily reliant on GPUs for computation. So as a project, we're working heavily on it, but we're not there sufficiently for this competition.
Okay, good. Any additional question? And just can maybe speak, ask, or just write in the chat.
I didn't put it. I guess I should have put it in the presentation. What I should have mentioned is that if you have questions related to GasNet or UPC++, you can. There's a UPC++ mailing list. You can ask your questions there, and the developers are very responsive. Generally, you'll get a response within a day to questions. So if you have some installation issue, or if you have some question about what some GasNet environmental variable truly does that you can't understand from the documentation, feel free to ask the GasNet developers or the UPC++ developers, the Pagoda team. And the best thing to do is to just post to their mailing list. You'll find it online.
And in general, I would say that if you have any questions, just feel free to open to ask us on the Slack, and we'll try to guide you to help with any of the questions. Okay, any additional questions? Okay, so I would like to thank Stephen. It was a very enhanced presentation, very nice. Nice to learn about the progress. And I will put the recordings and the slides, Stephen sending me later the PDF, okay? I'll upload that to the website so they can refer to that later on. And again, if you have any questions, feel free to contact us, and we'll happy to guide you and to find help. Okay.
All right.
Okay.
Bye, everyone.
Thank you, everyone. Bye-bye.
Bye.